7th seminar of DS academic calendar. Today, the speaker is the Dr. Maria Khalid. She is serving as assistant professor in University Institute of Biochemistry and Biotechnology. And the topic is the utilization of alien genetic diversity for improving drought tolerance in bread wheat. Dr. Maria Khalid will deliver the speech about 30 to 40 minutes. Then we have 5 questions. If somebody have the question during the presentation, can write in the chat room. Now, the mic is over to Dr. Maria Khalid. Madam, please continue the seminar.
Thank you so much, sir. Let me share the screen.
शहजाद साहब, डॉक्टर मारिया खालिद को स्क्रीन शेयर अलाउ कर दो।
सर जी अलाउ कर दिए।
जी मैडम, आपकी प्रेजेंटेशन अब ओपन हो गई है, शेयर हो गई है, प्लीज कंटिन्यू।
Thank you so very much, sir. So the title of today's seminar is Utilization of Alien Genetic Diversity for Improving Drought Tolerance in Bread Wheat. A brief introduction about myself. My name is Maria Khalid and I'm working as Assistant Professor IPFB at University Institute of Biochemistry and Biotechnology, Arid Agriculture University, Rawalpindi. I have completed my PhD degree in plant biotechnology from National University of Sciences and Technology. So in today's seminar, I'll be discussing a little bit about food security, more about the history of wheat improvement, the germplasm that I have collected to conduct my research, what are the advancement in molecular markers, in silico analysis to do mutational, to check the mutational analysis of drought tolerant gene, and at the end, I will discuss some of my achievements. So there is a myth going around that we haven't enough food in the world and hunger is a serious problem. It might be the case that your experience with the food is different, that you just have to go to, to the grocery store and buy the food stuff. But in fact, hunger is a serious cause of a death. We can say that hunger is a serious cause of a death after COVID nowadays, but it is serious cause of a death in different parts of the world, other than other serious diseases such as HIV and etc. So there are many factors that threatens our food security. Even your children and my children will have to deal with all the challenges like the climate change, like drought. So the world is still growing, and by 2050, we will need to extra produce 50 to 60% more food to feed an estimated 10 million population on this planet. So I think that the real challenge is we need to explore the genetic diversity and double the rate of genetic improvement of important crops, of our crops, such as in case of our country, as wheat is a staple crop of Pakistan, and other important crops as well. So let's move back to the long history of improvement with the larger impacts. When in the Neolithic period, the domestication of animals and plants occurred. Then in 1865, Mendel actually gave the concept of inheritance. In 1953, the Watson and Crick described about DNA, and in 1970, Borlaug introduced the dwarf genes. So that actually increased the 70% of the yield. So evolution of a modern wheat associated with the Green Revolution. So that thing happened in the International Maize and Wheat Improvement Program, known as CIMMYT. CIMMYT was established in 1966, and over the last 50 years, CIMMYT has developed the wheat germplasm with high grain yield plus biotic and abiotic stress tolerance. And one of the foundation known as the Rockefeller Foundation, they actually developed the Mexican semi-dwarf varieties. And further, they have distributed these varieties to various national agriculture research stations of different developing countries for wheat improvement. So the plant breeding method, that is a traditional plant breeding method, was being used for the selection of a traits that takes many, many generations of breeding and selection to develop the genetically stable crop that incorporating all the necessary traits so that it can take up to 15 years using the traditional approaches. Other than that, after the Green Revolution, what happened that the traditional farmer varieties were replaced by the modern varieties by continuous selection. So this results in narrowing the genetic diversity. And on the other hand, the mutation in bacteria and fungal population is quite rapid. So the population is increasing day by day, and on the other hand, genetic variation is decreasing with time to time. This became the scientific premonition when a canal burn epidemic was burst in 1985 due to just because of the narrowed genetic base. So scientists start thinking to interrogate the intergeneric hybridization. Then in 1985, the inter-wheat rye translocation occurred, and that results in fungal and insect resistance. But the drawback is that the poor is that of the poor yield. Furthermore, what scientists did is in 1998, scientists focused on interspecific hybridization. That is, they crossed the bred wheat lines with the wheat white relatives to form the synthetic hexapolite wheat. And then again, this synthetic hexapolite wheat is tolerant to biotic and is abiotic stresses, but they are poor in agronomic qualities. So they were further back crossed with the bred wheat variety to form the synthetic derivatives. So in NARC, National Agriculture Research Center, more than 800 synthetic derivatives were developed initially at NARC, but it was then reduced to total 171 lines for the better agronomic traits. So these 171 lines, along with 42 bred wheat lines, field trials were conducted for 2 years under two water regimes, that is well water condition and water limited condition. Eternal matter is used for the water limited condition to prevent the precipitation. And different agronomic, morphological, and physiological traits were recorded. And the details of this work has been published in our CSIRO paper. Furthermore, the genetic diversity on the basis of molecular marker has been studied many years ago. Basically, molecular markers are of two types, that is the neutral markers and the functional markers. Neutral markers are derived from the genomic region that is linked to the gene of interest, and whereas the functional markers, they are derived within the gene of interest. There are different examples of the markers, such as AFLP, rapid, SSR markers, and the SNP markers. Nowadays, SNP markers have been a marker of choice for the construction of a genetic map. But the main drawback of all these markers, basically these are known as the conventional markers, or and it is the time taking because it is a gel-based PCR we have to do. It is actually a laborious task. We have to load each of the sample and check whether if we want to do allele specific PCR, and we have to check that whether allele is present or not. So there is an advancement of other than the conventional PCR known as competitive allele specific PCR, which is a high throughput technique, but the uniplex technique, but the high throughput technique. So this competitive allele specific PCR is actually the fluorescent based PCR, in which two forward primers and a common reverse primer is used, and two types of fluorescent dyes used, and 2 rounds of PCR will be conducted, and according to the fluorescence, we can identify whether the sample is homozygous or the heterozygous. There is a general difference is shown in the slide of a gel-based marker with respective to the gas marker. So, for example, if a single person has to conduct a QTL study of 96 markers, what he has to do is, if he is using a conventional PCR, definitely it will take 38 to 50 days to complete the QTL study. But on the other hand, the gas marker, as the gas markers are of 384 well plate is used, or 1536 well plate is used. So in comparison with that of a conventional PCR, only 2 days or 1 day will be taken by one person to conduct the QTL study of 96 markers. In addition to that, maximum number of samples conducted by one person by using a conventional PCR is like 384 samples. But on the other hand, if he is using a gas PCR technique, or by using 384 well plate or 1536 well plate, total 18K sample it could be conducted within 1 day, or 73K sample could be conducted within 1 day. So basically, gas markers were developed by CIMIT CAS Beijing, China, that is Chinese Academy of Agricultural Sciences, one of the lab of CIMIT in Chinese Academy of Agricultural Sciences. And total, almost many gas markers, I think 140 or more gas markers has been developed by that lab. And that marker is not at all much costly if you will check the efficiency of these markers. And many other genetic and breeding programs and many other lab are actually using these gas markers. So we have also used the gas markers in our study. We used a total 131 gas markers on 87 functional genes. We have conducted this research on same synthetic derivative, which we did the phenotyping on these lines. And we have found some very interesting results. We have found some of the favorable alleles and some of the unfavorable alleles. These 87 functional genes are related to the drought tolerant, to the grain size, to flowering time, and to the end use quality related genes. And these red lines are actually showing the unfavorable allele, whereas the green lines are showing the favorable alleles. We have published all this research in Frontiers in Plant Science paper. We have also did marker trait association as well of in total 21 marker were associated with the grain or yield related trait as well. In addition to that, nowadays computer algorithms are very much used to check the mutation analysis. We have also conducted the similar work on one of the gene known as cell wall invertase gene. The main purpose, the cell wall invertase gene is basically an enzyme, and it has a source sink relationship, and it reduces the enzymatic activity under the stress condition as well. So main purpose of this enzyme, what that enzyme do is, basically, invertase enzyme are of 3 types. It is present in the cell wall known as cell wall invertase enzyme, those which is present on cytoplasm known as cytoplasm invertase enzyme, and the one which is present on vacuole known as vacuole invertase enzyme. So what happened that when sucrose food or in form of sucrose transported from source to sink via plasmidesmata, so the cell wall invertase enzyme actually converted the sucrose into glucose and fructose. Whereas in cytoplasmic enzyme also converted sucrose into glucose and fructose, and similar is the case with the vacuole as well. So what happened under the stress condition, drought stress condition, what happened? That it could not, the cell wall invertase enzyme could not perform their function very well, and it also affected the yield of a crop as well. The detail of this enzyme and another enzyme, that is a DRAP-D1, another gene, we have published in one of our chapter titled Analysis of Novel Haplotype Variation of DRAP-D1 and Cell Wall Invertase D1 gene that influence the drought tolerance in bread or synthetic wheat derivatives. We have given a overview about it. So what we have done by doing the in silico analysis, we actually selected some of the drought tolerant varieties and the drought susceptible varieties, and we cloned the cell wall invertase gene at the genome. Then we sequenced all the genes, and then we aligned all the genes in order to check if there is any mutation at the conserved domain known as the VEC-PDF domain. And we see, yes, there is a mutation in the VEC-PDF domain as well. So what we did is we used different types of a software. We did different in silico analysis in order to check that whether these mutations are synonymous mutations or these mutations are non-synonymous mutations. If these mutations are non-synonymous mutations, that means that they can affect the protein structure as well as the function of the protein. So some of the mutations were actually deleterious mutation. They can affect the structure and the function of protein. One of the software, that is protein variation effect analyzer software, we have used in order to check whether these mutations are deleterious or not. In my next slide, I'll show that one of the mutation, how it is deleterious or how it can change the structure of the protein. So in this diagram, one of the mutation, that is the aspartic acid, is changed to the another amino acid, that is a threonine. Both these amino acids have different in the function, and definitely the protein structure is also changed due to the change in amino acid. As you can see, the difference between their function, aspartic acid is bigger in size, threonine is smaller in size. Aspartic acid is negative in charge, whereas threonine is neutral. Aspartic acid is less hydrophobic and threonine is more hydrophobic. So that means that due to the difference in their properties, definitely there is a chance that the structure and the function of a protein will change. So this is the one of the example of the mutation. We have found total 7 risk mutation in this study that were either affecting the folding of protein or disrupting the stability of the folded protein. And we published our work entitled Characterization of Wheat Cell Wall Invertase Gene, which is associated with drought tolerance in synthetic wheat derivatives. Another germplasm we have collected from Kansas State University, known as International Literacy Mapping Initiative Population. And this population has been established by Mark Sorrells, and they actually crossed the drought tolerant variety synthetic hexaploid wheat with the drought susceptible varieties, that is Opata, and total 2043 recombinant inbred lines were produced. And these recombinant inbred lines were, we have collected total 216 recombinant inbred lines, and we did screening at seedling stage. We used cigar wool method to screen against drought stress under 2 water regimes, that is well water condition and the water limited conditions. And we have checked the root and shoot bar mass and root length and shoot length and various other agronomic traits. Furthermore, we have sent these lines to the Kansas State University for genotyping by sequencing, and the total 2,639 GBS marker were used to make the in the linkage map was constructed as well. From the linkage map, we have found some of the QTLs we have identified under well water and the water limited treatments. So a total 16 QTLs were identified, in which 7 QTLs were detected on 7B, position from 64 to 221 centimorgan. We published this result in Plant Genetic Resource entitled QTL Mapping for Seedling Morphology under Drought Stress in Wheat Cross Synthetic Opata. So overall, concluding from our this work, we have found some of the beneficial alleles for the genes that were fixed in the diversity panel with the frequency ranging of 96.4 to 100%. In some of the lines of synthetic derivatives, we have found only the favorable alleles that were fixed on the germplasm, and other than that, we have also found the 21 marker traits that were associated with agronomic trait under the both moisture condition, that is well water and the water level. Other than that, we have also identified from the ICME mapping population, we have identified 7 QTLs that is detected on 7B chromosome and that were positioned from 64 to 221 centimorgan. And from our in silico result, we have found non-synonymous mutation that were found in cell wall invertase gene at B genome, which is particularly on the conserved domain, that is the back-medium. So the future prospect of this work is favorable alleles for grain size and weight genes could be accumulated to enhance the grain yield in diversity panel, and QTLs that were detected could harbor important genes for drought adaptability. And we can explore the important drought-related genes that are still hidden on the 7B chromosome. In addition to that, what we can do is from our in silico work is we can do the expression analysis of cell wall invertase gene at B genome, which is required to analyze the effect of mutations on the function of gene, that is cell wall invertase gene at B genome. In addition to that, we can do a collaborative work with the plant breeding and genetics department, with the agronomy department, and with the computer science department as well, in order to conduct a high throughput phenotyping technique for the advanced genomic studies, as well as some in silico analysis, some other techniques like the machine learning. If you want to learn any language like Python and statistical language like RStudio, so computer science department is the best department to teach us all these things as well. And as agriculture, there is a many advancement in phenomics and in genomic studies as well. So we really need a bioinformatician to analyze all our statistical analyses as well as our machine learning approaches as well. I have one of the project funded by AGC at University Institute of Biochemistry and Biotechnology, titled Exploring Genetic Diversity and Association Mapping of Maize to Analyze Root System Architecture under Well Water and Water Limited Conditions in collaboration with NARC. My another project is going on entitled High Throughput Phenotyping to Analyze Root System Architecture on Synthetic Derivatives in collaboration with Dr. Lee Hickey from University of Queensland, Dr. Vas Rashid from Kailasm University, and Dr. Alina Gul from NAST. My third ongoing project is going on entitled the Hyperspectral Estimation of Biological Nitrogen Fixation in Dry Beans in collaboration with Dr. Eddie Thompson from Michigan State University. These are some of my achievements. I have won different scholarship, and Michigan State University, USA actually sponsored me to do a postdoc there, but unfortunately I didn't awarded by the degree. That is why I worked there for 6 months as a visiting scholar. Then I've also won AirSIP, and I went to University of Queensland, Australia, and worked with Dr. Lee Hickey for 6 months. Then I won a TÜBİTAK scholarship for 1 year, and I went to Sabancı University, Turkey, and I worked with Dr. Hikmat Budak. Then I've also won finally the Food Science Security Scholarship, which is a German-based scholarship, and I went to CIMMIT lab in Beijing, Chinese Academy of Agricultural Sciences, China, and I worked with Dr. Chumbuha for 6 months. These are all my publications, other than the publications I've mentioned within my slides. These are my chapter publications. I've also presented a poster in 2 conferences. One is the annual Illinois Corn Breeders School, and another conference is Maize Breeding 2019 at Missouri State, USA. Where I presented a work of hyperspectral estimation of biological nitrogen fixation in dry beans. Where I did some of the machine learning work there. These are all my mentors. My supervisor is Dr. Elvina Gul, Assistant Professor Asap Nas. My co-supervisor is Dr. West Rashid, Assistant Professor, Qaid-e-Azam University. Dr. Chumbuha, who is a distinguished scientist in CIMMIT CAS, China. Dr. Lee Hickey, who is Associate Professor, University of Queensland. Professor Dr. Hikmat Budak from Sabancı University, Turkey. And Dr. Addie Thompson from Michigan State University. At the end, I would like to thank to the respected Vice Chancellor, who actually gave us the opportunity to all the faculty members of different departments to present their work. And I'm very, very much thankful to Director of Advanced Study, who actually created the platform for us to present our work. I'm thankful to the Director, Dr. Ghazala. Thank you so much, ma'am, for choosing me to present my work in front of all of you. I would like to thank to the faculty member, Special Dr. Javed, who took his precious time and gave me suggestions to improve my presentation. Thank you so much, sir. I would like to thank to Dr. Elvina Gul and my supervisor and Dr. Awais Rashid. Because of them, I traveled the world and gained different experiences from different institutes. I'm really very thankful to them. And at the end, I'm thankful to all the scholarship programs and especially the Michigan State University, who actually sponsored me to work there. Thank you, all of you. Thank you very much.
Thank you, Dr. Maria Khalid, for presenting a wonderful seminar. Today's seminar. If people have a question, the house is open. We can take 5 questions as suggestions.
May I ask questions?
Ji, please.
Dr. Maria, can you explain how many wheat accessions are there in the Pakistan or in the world?
Thank you so much, sir, for your question, but I cannot answer, like, I'm really not sure about the total how many accessions are there in Pakistan or in a whole world. But what I've worked in synthetic derivatives, total 171 lines are there at NARC. And I've worked there, and the ITME mapping population, total 2,043 total recombinant inbred lines are there, in which I've worked on only 216 lines. Thank you.
Do you think that the genetically engineered crops are more successful as compared to others? Conventional?
Well, sir, that is actually a very debatable question, and still debate is going on. But I'm totally okay with the GMO as well and with the plant breeding method as well. Both methods are in equal importance. Nowadays, even the speed breeding is the advanced technique to speed up the breeding the generation as well. So every breeding methods or the conventional or non-conventional methods has their advantages and advantages as well. So I'm okay with both of the, the genome editing is another technique, which is a very good technique. Conventional breeding technique is a very good technique. And all these techniques, the main purpose of all these techniques is to improve the crop or to increase the yield. The main purpose is that.
Mhm.
Thank you.
Thank you.
Madam Ataula is speaking here, PhD scholar, plant breeding and genetics.
Yes, please.
Madam, you use marker in your study or only these advanced techniques like SNPs and other things?
I did work on the GASP markers, which is competitive allele specific markers on the functional genes. These markers is actually present within the functional genes. Other than that, my other work is on GWAS, that is genome-wide association studies as well. We will publish that work soon, inshallah. On the other hand, my work is on some of the machine learning work as well on the ribenes as well, and I did some work on maize crop as well. And basically my work is on genome-wide association studies or the GASP markers. Thank
Where you, madam, search these markers? These are reported markers, are available any repository, or where you found these markers?
Yes, these are all the reported markers. These are actually developed by Summit GASP, where I have been there and to do my research work as well. They actually developed these gas markers. And nowadays there is an advancement of a gas marker that is known as the START markers. Basically that Beijing lab is doing work related to the gas markers.
Thank you, madam.
Well,
Next, please, if you have any question. I think no more question from the audience. Thank you, Dr. Maria, for a wonderful presentation in the advances in science about the molecular technology you used in your publications. One thing very important, I request the chairpersons and deans and directors, please guide your students to join these seminars, because we are here for the guidance, for cooperation with the students, to teach them, to tell them advanced technology. As just like today, the speaker is from the UIBB, then must be UIB students, plant breeding students, agronomy students, and should be there. But still, today is about the 60 participants attended them. I will request the chairpersons, dean directors, research coordinators, please communicate with the students and guide them to join the seminars with their relevant subjects. Our purpose for the seminar schedule that can be helpful for the students, as the speaker will guide them, will teach them the new technology. Thank you, all participants, and thank you, Dr. Maria, for sparing your valuable time for this seminar. And today the seminar is ended. Thank you very much.
thank you, sir.